Okay, so taking a step back here and looking at what we have covered. Remember, we have an exam coming up. When is it? Yeah, it's Tuesday, a week from now, right? All right. So I got hot now. Okay, so activating mitochondria, right? By the way, cold exposure is good for activation of mitochondria. All right, so what is going to be on the exam, the things that we have covered and the things that we need to cover, right? So just to review here, things that we have been talking about on week 2. So we talked about here the 2 types of hematopoietic paths, right? We talked a lot about the myeloid cells and lymphoid cells. And here we started talking briefly about those functions of those cells. And we are talking about innate immune system. We briefly talk on adaptive immune system having memory, right? And then we talk on some types of dendritic cells, 3 different types of dendritic cells, where you find them, what are their major functions, right? Now here we started talking about your phagocytic cells and how is it that they get to work. And we talk a little bit, I don't think we talk much about adhesion molecules. We'll talk a little bit about chemotactic factors and how those phagocytic cells like to go towards higher concentration of these chemotactic factors, and that is called chemotaxis, right? And here I briefly mention a little bit about phagocytosis and phagolysosomes. And today we're really going to cover details on those pathways, okay? And then we talk a lot about innate immune responses, but we usually put this in a big bucket of not having memory type of immune responses. And then we talked some about those pattern recognition receptors. Remember, we have about 100 genes or so that will code for those pattern recognition receptors. And overall, you can identify about 1,000 different patterns, 1,000 different molecules, okay? And then we still have to cover these parts here, and this is where we're really going to focus today, okay? One covering still wrapping up here on the week 2. So we have to look at ways of phagocytosis and elimination of microbial microbes, right? I'm going to look at oxygen dependent and oxygen independent pathways, right? And then we are going to look at general activation related to signaling, cell signaling from those PRRs. So we are going to talk here about your NF-kappa B pathway and activation. Okay. Then we're going to talk about this very, very important inflammatory pathway. And this is also due to receptors of PRRs activations. And this is going to be your NLRP3 signaling. Okay. This is really, really cool stuff. Okay. So just to make your lives easier, I added these 2 podcasts for the lecture 4 videos. I noticed just a few minutes ago that 6 of you had actually looked at it. Not sure if you watched the whole thing, but you looked at it. That's good. Okay. I did take a lot of time and dedication to get this thing ready for you, and I really like the way it came out, like really giving you this broad aspect of those responses, right? So this first podcast here is talking about those PRRs, right? And we do have some slides here for this lecture that I took from there. And this one here is the PRRs, and this other one here is talking about those lymphoid cells and the type of immune response that you get based on activation of those lymphoid cells, right? So I know that's a lot. That's why we had the exercise, right? Okay. So here, after we wrap up with, yes? Sorry. Okay, so did anybody have trouble with the Jove videos? Is not going through at all? Yeah? Okay. Oh, okay. So you do have a way of signing up for it. Oh, okay. And that's another study strategy that I forgot to mention right in the beginning. I really encourage you to form study groups, okay? Because we don't have SI for this class, right? And if you get the material and form small study groups, I say, like for my last two classes here, what I used to do is like stay after class if you have the time. If you don't have a class, like right following this one, right? Does anybody have a class like right after this class? One person. Okay. So what about just before this class? More people. Okay, so you have to figure out how you're going to do this, how you're going to do the study groups. So if you can stay right after the class, I'll totally encourage you to do that. Okay, and actually, I think Tuesdays and Thursdays, I think my son has class until 6 o'clock or so, so I can hang around for another half an hour or so and try to direct some study groups, if that will help, if you guys want to do that, okay? Yeah, that really helps. So I would say with this, yeah. Yeah, that's the thing. I have been putting practice questions, practice quizzes with the Jove videos. So first you have a list of Jove videos, and then you have the quiz. And if you do not go through the videos before the quiz, you can actually watch the videos within the quiz. But I'm not sure if that is practical because I do think you have some time limit in there. I don't remember now. So yeah, actually with the Jove videos, most of the questions I wrote them. It's not that they are there. I don't. Sometimes I don't like the way they put the questions, so I modify the questions for the practice quizzes. Yeah. So yeah, it's a good way to to get used to the information and really focus on what you need to. Okay. Yeah, you can do that too. Yeah, with your GeoVis, you can do your own test. But with that, I am not sure if it will populate the original questions. So I would stay away from there because if it populates the original questions, it's not really focusing on what I want to focus. And also, I found a couple of mistakes that it might not be too helpful. But yeah, but you can try that, just in case. If you start getting confused, don't try that. Okay. All right. So that's my my advice to you. Make sure you get into study groups to really understand what's going on. All right. Okay. So theoretically, the learning objects for this week, they're really to focus on inflammation, okay, everything to do with inflammation, including acute and chronic inflammation. And here we should cover the complement system, right? So if we can cover this and then be done for the week, I'll be happy. And the part for antibodies and antigens, this is actually not very complicated. So I might do just like I did with last term, putting in a self-study videos and basically just go over parts that are to be more emphasized. Okay. All right. So picking up where we left off with the innate immune functions, right? So we left off on this slide here. So we're mentioning that your innate immune system, your pattern recognition receptors, they are designed basically to detect parts of microbes that are essential for the microbial life. So that ensures that in case the microbes try to mutate those parts or get rid of those parts, the microbe itself would not be able to survive. Okay? So, for example, here, when you look at PRRs like TLR3, TLR7, TLR8, those are going to detect nucleic acids that are very important for microbial genetic information and replication. Okay, so in this case, for example, TLR3 is going to detect, look at this, double-stranded RNA type of structure. So this TLR3 is going to look for characteristics that are not very common for human cells to have, for things that human cells are not going to have in certain places, all right? So if this thing is detecting viral RNA, either double-stranded or single-stranded, then the virus cannot really get rid of the RNA. I mean, that's part of the genetic material, right? So this type of detection is fundamental to start a reaction that is eventually going to get rid of the virus, okay? And then we have, I was going to see here, some other type of PRRs that detect a type of DNA that is common for bacteria, but is not common for human cells. Okay, so in this case, this PRR detects a type of RNA that doesn't have methyl group on those CpG islands type of thing. So that means when it detects the DNA without that methyl group, it's going to think, oh, okay, so this must be an invading organism. So let me start activating antimicrobial genes, and let's get rid of whatever microorganisms around here, because this type of DNA is not supposed to be here. Okay. Then you have certain PRRs that are going to detect certain compounds or certain molecules that are totally not present in human cells. Okay, because you can say, well, sometimes you have double-stranded RNA that are, normal type of conformation in normal cells, but then sometimes you have certain molecules that you totally do not find in normal cells. In the case here of lipopolysaccharide, okay, LPS, this is really a component of gram-negative bacteria. And you're not going to find it in normal cells. You're not going to find it at all. So when you have LPS that is detected by PRR, the response to the presence of the LPS, that response is going to be a very strong response. So LPS is a very strong inflammatory type of trigger, okay? Okay, so on and so forth. So this is only words. We're going to see in more details how that is happening. Okay, so types of those receptors and where you're going to find them, right? So type of PRR, you're going to have receptors that are inside of cells. So that means they can be in the cytoplasm or they can be inside little vesicles, right? Endosomes, okay? You have PRRs that are on the surface of cell, on the cell membrane, okay? And you have PRRs that are just floating around in the humors, like on these fluids, right? They can be in the blood and the lymph, and they're just secreted. They're just out of the cell. They're floating around, okay? So the main idea here, the main concept is, why are these PRRs in these places, okay? Why are they located there? And what is their function? What is it they're doing over there? Okay, so we're going to look at this and say, all right, if a PRR is on the surface of the cell, it is going to detect some type of microorganism that is extracellular. What kind of microorganism that you think it can find there? What can you find that is a pathogen that is outside of the cell? Bacteria, fungi, viruses, right? Something that is not inside of the cell. Okay, so that makes sense. Now, if you have a PRR that is in the cytoplasm, what is it going to detect? Intracellular pathogen, right? So let's say here, for example, that you had a virus that was floating around here, but avoided detection by a cell surface PRR, and it was able to infect the cell, and now it is inside of the cell, and it's thinking, oh, I really snuck inside of the cell, and that guy did not detect me, and party time. It's like, no, there is another PRR that is going to detect the cytoplasm of the cell. So there's another chance of catching that virus that was not caught when it was outside of the cell. Okay. With the same line of thought, you can have viruses that get engulfed and end up in lysosomes or in endosomes, and then you have PRRs that are going to detect the virus in those endosomes. Okay. And then you have these guys over here that are just floating around, and they're really looking for anything that they can bind to. Okay, so they're going to be just patrolling all these fluids, and just in case, you know, you have some bacteria that are not really getting into contact with these surface PRRs, these guys here should be able to catch them before they cause trouble. Okay, so that's the main idea here. What is the PRR doing where it is located? Okay, and do they have a backup system? Okay, so, oh, I didn't remember that. All right, so here, location of those PRRs and what is it that they are targeting. So first we're going to look here at those soluble PRRs. They're just floating around everywhere. And yes, there are lots of them, right? So we have here something that is called pentraxins. You have colectins. They look like a little spaceship. And kind of looks like these other guys here, ficcolins. Ficcolins and colectins, they still look like this spaceship kind of thing. So where you're going to find them in the plasma, in the blood plasma. Okay, this is not cytoplasm. Okay, this is blood plasma. Okay, and they are working around, and what is it they're going to interact with? So most of them are going to interact with PAMPs, right? And some of them interact with DAMPs. Actually, C-reactive protein is a DAMP, so it's kind of in the wrong place here. So let's see here. This guy here, colectin, is going to is going to detect sugars. So you have the one that is called mannose binding lectin. So mannose binding lectin is really a type of colectin. Okay, mannose binding lectin is going to bind like what kind of molecule? Sugar. In specific, what is the sugar? Mannose is in the name. Mannose binding. Lectin is the type of molecule. This one is going to bind mannose. Okay, so binds carbohydrates that have mannose in them. Okay, and also binds here like fructose. Now, one thing that is easily not seen when you're going through, through the book and through the literature about these types of floating PRRs is how is it that that they get to do their job, okay? Because sometimes, well, all the time, you're going to find carbohydrates on the surface of normal cells, right? Have you seen that? Do you have carbohydrates on the surface of normal cells, like eukaryotic cells? Yeah, we have mentioned even last time that we had sugars on the surface of red blood cells, right? So you have these carbohydrates on the surface of cells, but then look at this. When you have here this eukaryotic cell, okay, so we put some, I don't know, let's put some carbohydrate here. These are carbohydrates, okay? All right. And then you come with something that is a colectin, or a colectin is floating around over here, and it looks like this little spaceship that has legs. Okay, the legs are the ones that bind to those targets. Okay, so then this thing is floating around, and it finds the sugar on your own cell, and you say, wait a minute. So if I have the sugar on the cell, am I going to activate inflammation? Am I going to activate destruction of the cell? And the answer should be no. Why? Well, you see how many legs you have in these things here? In order for these molecules to get activated and start an inflammatory process, in order for them to do that, they have to bind to at least 2 of those targets. But those targets, in order for that to happen, these targets, they have to be in close proximity on the surface of a target cell. Okay? When you look at a normal cell, you can find those patterns, but they are not close together. They are not in high density in one spot. Okay? Now, if you compare that to a bacterium, so here is the bacterium, and here is the circular DNA of the bacterium, not the nucleus, right? So here's a bacterium, and now you look at those sugars on the surface of the bacterium. They are everywhere on the surface of the bacterium, okay? Now, if you get those PRRs, get a little spaceship here with those little legs, okay? So you get those PRRs that interact with targets on the surface of the bacterium. Now you're going to see at least 2 of those get to interact, and that interaction causes a change in the conformation of this big protein over here, because these guys are proteins, okay? So it causes a change in the conformation of the protein, and that is going to trigger a response and activation of the alarm system for inflammation, okay? So the density of those targets on surface of cells, the density is really important, okay? So it's not like our cells don't have those target molecules. It's just that our cell doesn't have a lot of those target molecules. And that's one safety mechanism of not activating this type of inflammation against cells that are just your cells, okay? So this is an important concept here. All right, yeah.
So when like the colectins attached to like the normal eukaryotic cell, what happens if they recognize that this is
so this is it. So let me okay, imagine I'm collecting, okay? So I have 4 binding sites, 2 arms and 2 legs, okay? So I'm gonna go and I'm gonna say, okay, this is normal cell. I go there, I recognize this, I attach here. So, but I only recognize one of them, I don't attach anything else. I say, okay, I'm not gonna do anything with it. It's not crazy glue. It's attaching, get interaction, no reaction. I'm gonna float away, I'm gonna look for something else that I can actually interact a little better with. That's a bacterium right there, and I'm floating around, mind my own business and all that stuff. Now I'm stuck. Now I got 4 interactions, okay? When that happens, I mean, obviously I was walking around and then this happened, my whole body changed. Now the change of the body is gonna send a signal that is gonna make something happen. So the whole thing, change conformation, is gonna trigger other stuff to cause inflammation. It's gonna start, producing, that's the part you're gonna get next, okay? So density of those target molecules is really, really important for activation of those soluble PRRs. Okay. All right. Okay, so there are some of these PRRs that are very important for activating the complement system, which we don't get there yet, but we will, right? So what are those PRRs? You have here PRRs that fall into pentaxins. They have 5 legs to them, okay? And one example here is C1q. That's the name of the PRR, okay? It's a complement 1q protein. You have these guys here, the colectins, okay? So mannose binding lectin is a very important one. And ficolin, those are all under this colectin kind of thing, okay? Colectin 11, all these guys here. And okay, ficolin actually is a little bit different. They're all binding these sugars, right? And then you have this other guy here that is a protein called propyridine, and that is also going to recognize the stuff on surface of pathogens. And this guy is cool because it can also recognize LPS. Look at that, all right? So propyridine is floating around and looking for gram-negative bacteria, okay? So these are examples of those PRRs that can activate a complement pathway. And when you get to different paths or different routes of the complement pathway, then this is going to start to make more sense, okay? All right. So here, besides those, you also have molecules of the complement system that work like they are PRRs, okay? So as you're going to see with the complement system, you have these proteins floating around, and they float around in this pro form, in this zymogen form. They are not active, but if they get cleaved, they become active. So when those complement proteins get cleaved, the one important one is C3, okay? You're going to have C3 that is going to get cut in C3a and C3b. So C3a is going to float away, and then C3b is going to bind to the surface of pathogens. So those complement proteins, they are going to function like PRRs, recognizing patterns or recognizing surface of pathogens, okay? So yeah, everything is very superficial right now, but we're going to get into more details, and this is going to make more sense. All right. So here, more details on how those PRRs work, right? So here's when you have to get some memorization, right? So localization and function of those PRRs. First, we're going to look at the ones that are on the cell surface, okay? So those are extracellular, but on the cell membrane, okay? So here you're going to find TLR 1, 2, 4, 5, 6, right? 1, 2, 4, 5, 6 on the surface of the cells. And then you're going to say, okay, if you have only one PRR over there on the surface, what is it doing? What is this molecule really? And you look at it, it has a C-looking binding site over here, right? And then it goes through the membrane, and it has an intracellular domain over there, okay? So by itself, without being bound to anything, this thing is quiet. When it binds to something that it recognizes, then this thing is going to get activated. But it's not going to get activated if it only binds alone, by itself, to a target, okay? It needs to dimerize, okay? So most dimers of those surface TLRs, most dimers are homodimers, so you get dimers of the same thing, okay? So this guy here will have another orange thing that looks like that to do the dimerization. But then some dimers, they can be combination of different things, like TLR2 and 6, 2 and 1, and etc. Okay? So depending on the combination of those dimers, they are going to send what type of signal to the inside of the cell to tell the cell to start fighting that pathogen. You're going to start telling the cell, look, you got to produce some cytokines that are going to kill the pathogen, okay? Remember, what kind of cells have the receptors? These PRRs. Here they are. You have macrophages, dendritic cells, neutrophils. These guys here, are innate cells, right? So these guys are your innate cells. Innate, all right? And then you have these other cells here, look. Monocytes, okay, part of innate cells. Epithelial cells. What are epithelial cells? On the skin. What about endothelial cells? Inside blood vessels, veins and arteries, endothelial cells. Okay? So those cells, they also have PRRs. Okay? Now, a couple of them that you should remember, okay, is Toll-like receptor 4, okay, so TLR4. This is a very important one that can detect LPS, so that is your gram-negative endotoxin, LPS, right? And then you have this other one that is TLR2, okay, so 2 and 4 are on the surface, okay? And that one detects a gram-positive LTA. Okay, so you have 4 that are for negative vibes, and then you have TLR2, those are for positive vibes, positive gram-positive bacteria. Okay, what they are detecting here. All right, so here I put a little thinking brain over here, right? So I say on the cell surface, they have to form dimers in order for them to get activated. Oh, something else about the activation. When you have dimer formation, the intracellular part of those receptors, they interact. If they're floating around by themselves, you don't have anything interacting down there. Okay. But when they dimerize, you have this interaction in the intracellular part, and this is a mechanism that sends a signal to the inside of the cell that something has to be done. And this is actually really cool because depending on the dimer that you form, that is going to tell the cell what kind of pathogen that you have seen. Okay, so you have like 2 and 4 that can detect gram-positive bacteria. Okay, so that is going to tell the cell upregulate genes that are going to fight gram-positive bacteria. Okay, but if you have a dimer of 2 and 1, that gets activated by patterns that you find on fungi. So this interaction is going to tell the cell upregulate genes against fungi. Okay? Remember, if you have bacterial infection, and these are your innate cells, bacterial infection, they are going to activate a pathway that is going to be what type of immune response? Type 1 against bacterial infection, right? But it could also be inflammatory, type 3. Depends on the bacteria, okay? So let's see if this is extracellular bacteria, that's going to be type 3. If it is intracellular bacteria, that's going to be type 1. So let's say this guy here is outside, is gram-positive, and it got detected. So now this cell is going to do inflammatory type of response. That's going to be a type 3 type of response. This guy here, let's say also fungi, is going to be type 3 type of response. So on the surface of the cell, you are going to have mainly type 3 if you have fungi or bacteria, right? Okay, so let's see what's happening here. On the cell surface, important receptors to know, again, is TLR2 for LPS, TLR2 for gram-positive bacteria, 2 positive, TLR4 for LPS, that's gram-negative, okay? And, okay, TLR5 I put there, but I usually don't even ask about TLR5. I ask 2 and 4, because they're easier to remember. Okay, so now, looking at intracellular stuff. So, inside of the cell. If you look at these receptors that are bound to membrane inside of the cell, that is going to be in endosomes. Okay, so these are the guys that are going to detect viruses that could penetrate the cell, okay, or engulfed by the cell. Okay, so now Now let's see what happens here. You have certain TLRs that you find in endosomes, and they are here, 3, 7, 8, 9, right? Remember, on the cell surface you had 1, 2, 4, 5, 6. Now on endosomes you're going to have 3, 7, 8, 9. Okay? So what are these guys going to detect here? Well, they're mainly going to detect either RNA or DNA, okay, of the pathogens. Now, usually you are not going to see RNA and DNA of viruses or bacteria, okay, just floating around in these endosomes. Well, because these pathogens, they are hiding the RNA and the DNA. They are hiding it inside a capsule. So they have the genetic material protected from detection. So these guys here, they want to look at DNA and RNA, and they can't see anything because everything is inside this protection capsule. So something else needs to happen here in order to expose the inside, the guts here of those pathogens. What do you think is happening there? Well, they're already being phagocytosed, because if these guys here are inside of an endosome, it could be a phagosome, right? They were out on the cell surface and they got inside of the cell, so they're inside this little space here, okay? But what happens here now, okay, let's see, the cell is a phagocytic cell, and it gobbled up these microbes, these viruses, and it has the receptors that can detect RNA and DNA, but then nothing is going to happen because the DNA and the RNA are protected inside this viral capsule. So one thing that the cell needs to do here is to break open that capsule. Okay, and one strategy that the cell uses is by pumping hydrogen or hydronium ions inside of this endosome and making the environment here really, really acidic. Okay, so you have a proton pump on the surface here of this little vesicle. Okay, so you have a proton pump here that uses ATP and pumps protons to the inside of this cell, of this vacuole or this endosome, and makes it really, really acidic. Now, when this place gets very acidic, that is going to break open this protein capsule of viruses. This is how the DNA or the RNA gets exposed inside of this endosome. Okay. Then once the RNA or the DNA are out of there, then these PRRs are able to detect it. Okay, it's a very, very clever mechanism, because remember that before these PRRs were in the endosome, they were out there on the cell surface, but out there on the cell surface, they were not really working, right? Because you're not going to find viral RNA floating around, right? Because the viral RNA is inside the capsule, and over here you're going to have virus capsules floating around. So you have to think, where is the target in my pathogen? Is it on the cell surface? If it is on the cell surface, then those guys will be able to detect it. But if it is inside of the pathogen, like those nucleic acids, now you have to really acidify the stuff, break open the pathogen. Now you're going to have PRRs to look at them. Okay, so important PRRs to know here. You have TLR3. Okay, so this guy here is important for detecting double-stranded DNA. And you have TLR7 for single-stranded RNA for RNA, okay. 7 and 8, both of those are single-stranded RNA. And then you have this cool guy here that is TLR9. So this guy is going to detect your CpG unmethylated DNA. Okay, and that's mainly for bacterial DNA. Okay, and how you're going to remember TLR9 detects cpgDNA. Look at this. This P here is kind of a mirror image of 9. That helps you remember TLR9 detects cpgDNA. Okay, so field detection things here. All right. So here, looking at those endosomals, okay, TLR3, double-stranded RNA, TLR7 and 8, single-stranded, TLR9, bacterial, unmethylated, cpgDNA. And then you have these other guys here, look at this. In the cytoplasm, floating around the cytoplasm, not in endosomes. You're going to have NOD1 and NOD2, okay? You're going to have RIG-I or RIG-1, and you're going to have this guy. This is a big one, NLRP3, okay? This is also part of the NOD family, okay? And this guy detects the bad guy over here, the LPS. Okay, did you have something else that detected LPS before? What was it? 4, right? So TLR4 on the surface of the cell that is capable of detecting LPS. But now, let's say that TLR4 did not did its job, and the bacteria, LPS is for gram-negative bacteria, the bacteria got inside of the cell, and it is an intracellular bacteria. Now you're going to have NLRP3 that can detect LPS. So that's a backup system in case your surface molecule failed. Okay? And this guy here is a very, very important one for activation of inflammation. That's what's going to activate the inflammasome beast. Okay? All right, so finally here, one question coming up. Okay? So toll-like receptors found inside an endosome would most likely recognize what?
Yeah.
Does anyone need more time? So, okay, I'm going, oh. Okay, some people are changing their minds and giving me the wrong answers. No, you're going the wrong way. Okay, change your answer back. Whoever, no, okay, I'm gonna stop it. All right, here we go. What is your answer? A. All right, so now you really have to think here about what is the endosome. That is your little nucleus. The endosome is right here, right? What are you gonna find in the endosome? You're gonna find the guts of some viruses or bacteria that got open up because of acidity, right? So you're gonna find here what again? Single-stranded RNA. What about this? What about chitin? This is chitin, right? What is chitin? Sugar on the surface of some fungi, fungus, right? So if this is sugar on the surface of a fungus, then you don't need this intracellular PR to detect it. LPS on gram-negative bacteria, that's on the surface of the bacteria. Okay, so it's not going to be detected by endosomal stuff. And same thing goes with that. Flagella on bacterial cells is also on the surface, right? Okay. Do you have any cells in the human body that have a flagella? Yeah, what is it? Sperms. Would that be a problem if you have a PRR that detects sperms? Yeah, all right, let's go for a vote. How many people think yes? Okay, yeah, you gotta vote. Okay, yes. How many people think no? It actually is a big problem. Because if you have innate immune system that is able to detect the flagella and kill the cell that has the flagella, what's going to happen to all the sperm? They're going to die. So it is a problem. How do you get around this problem?
You tag it like for your own, like doesn't it have a tag that your body recognizes?
How do you go around that problem? Why is it that the woman's body doesn't kill all the sperm before fertilization?
You just have to do it in vitro.
Everybody has to go through in vitro
No.
fertilization. That would be expensive and not as much fun.
Don't all the cells die before they make it? Very few, like in a function.
And then whole humanity is going to vanish. I mean, all reproducing beings. That's going to be a big problem. How did nature go around this problem?
They have like maybe a protected chemical on their surface.
You were right when you started protective part of it.
Protected capsule.
No.
They have like fructose or something on.
Oh, they do have a lot of fructose because
Yeah.
they need fructose for the energy. They have to swim really, really
Yeah.
good fast. Ah, you see the kind of problems that the immune system have to deal with? These are not easy problems. They don't have easy answers. But the immune system is very clever. It figured out that it should not attack the species sperm, right? So it designated some organs as immune privileged organs. Reproductive organs are immune privileged. And so is your brain and your eyes. Okay, because you do not want PRRs and your innate immune cells attacking stuff that they can recognize over there. And this is why, and this is how animals don't go extinct. It's a good idea not to attack, foreign sperms in case you want to reproduce. Right? And then if you think about it, when a woman is pregnant, you are carrying a baby that only has 50% of the genetic material that is yours. So 50% of the genes of this baby, they are foreign, is not yours. Why is it that your immune system doesn't attack the baby and kill it? Well, first, that would be a very bad idea. You're going to go extinct, right? Well, because that's a site of immune privilege. You're not going to have immune system attack at those sites, right? So it's not like the immune system, it's not like these PRRs don't have the receptors on your normal cells. It's just that sometimes the immune system has to come up with very ingenious ways in order not to kill you or the babies. Okay, so it has to think about a lot of stuff if you think about it, right? All right.
But they would attack like if the mother's like RH.
Yeah, that's a different story. We'll get
Okay.
there. Yeah,
Okay.
yeah. But yeah, we have all these problems to deal with.
Yeah.
Immune system has to decide what is self, what is non-self, and if something is non-self but is useful, that you should not attack it. What you're going to do in order not to attack it? Yeah.
Is maybe a cause of infertility the immune system attacking sperm? Or is it different?
Yeah, that is a cause, but it's very, very rare. There are some women, picture this out, maybe I should not record it, but that's a problem. They could not conceive because of allergic reaction. Okay? Overactive immune system attacking things that it should not. Okay? All right, so back here to see PRRs can get really interesting, right? Okay, so here. PRRs that attack damage-associated patterns, right? So look at this guy here. This guy here is called rage, okay? Rage, not that he's angry, it's like receptor for advanced glycation and products. Where is it you're gonna find the glycation and product? Like when the cells get older, they start to glycate sugars. When you have case of diabetes, either type 1 or type 2, and you have elevated blood sugar for a long period of time and it's not very well controlled, you're going to get a lot of glycation products. In that case, you're going to activate the rage. It's going to be really mad about sugars, more sugars than they were supposed to be there, okay? And they can start to attack those cell damage patterns and cause inflammation. Because remember, two main functions of innate system is production of inflammation and antiviral response by interferon, okay? Via interferon. Okay, so rage is a problem. And then you got this other guy here, NLRP3, okay? This guy can recognize deposits of little crystals that are made of uric acid. You know when that happens? Gout. Everybody heard of gout? Very painful, inflammatory condition. How do you get gout? No, poor diet like that will give you type 2 diabetes. But gout you get if you eat a lot of animal products. It used to be called the king's disease or something like that. A lot of like salmon, red meat, and things like that, okay? Because gout, uric acid, you get with the breakdown of purines, purine breakdown, okay? And you're gonna find purines where? Purines and pyridines you find in nucleic acids, yeah, in animal muscle, fleshy type of thing. So you look at the red meat, and you're gonna find that's muscle. Each muscle cell has a lot of nuclei that is packed with nucleic acids, right? So if you compare that to a plant protein, you're not going to have that problem. But if you eat a lot of protein, a lot of animal-based protein, you can have deposition of uric acid crystals. They can accumulate. Crystals are heavy. They go into circulation and they get to accumulate in the parts that are not very favorable for circulation. Like, you know, they're heavy, they go down to the feet. So where do you get gout? Usually in the joint in the big toe. Because it goes down and it doesn't come up very easily, it gets stuck in there, and then you get recognition of gout by of uric acid by NLRP3, and you activate this very crazy inflammasome system. Okay, lots of inflammation.
The only thing that comes to mind
Asparagus? I never heard that. But
so even some vegetables
okay, yeah. I never heard of asparagus. Yeah, if you're starting with kidney failure, then definitely you have to control the amount of protein, especially animal protein. But yeah, I don't know about that. I'm gonna look it up because it's interesting. I just never heard of it. But you should definitely, you know, slow down with animal proteins in case of kidney failure. Yes. All right, so here, let's go now to the mechanisms, okay? This part is gonna get more interesting. So how is it that, again, you're stepping back, and how is it these receptors are working? First, you have to recognize the thing that it has to bind to. Then you're gonna activate a pathway of cell signaling that is going to send a message to the nucleus of the cell and is going to cause some kind of response, okay? And some other times you're going to activate proteins that are in the cytoplasm that they're just waiting to get activated, right? But they're already present there in the zymogen form, on the inactive form. So once you have these responses, you can produce certain cytokines that are antimicrobial, and then you can get rid of the pathogen. And also you're going to produce inflammatory cytokines that can get rid of the pathogen. Okay. All right, so here, the mechanisms that you need to know, okay, four of them. They're focusing on these three mechanisms here. The other one is just a signal of gene upregulation. All right. So first one, we're going to look at phagocytosis and oxidative stress, oxidative damage, respiratory burst. Then we're going to look at NF-kappa B, then NLRP3, and this one is actually easy. That's like a piece of cake. All right. So here, what are the PRRs that are going to trigger phagocytosis and respiratory burst? Okay, so those, of course, they have to be on the surface of your cell, okay, because if it's going to trigger phagocytosis, it has to bind something and eat it, right? So it makes sense that it's on the surface of the cell. And those are your lectin receptors that could be floating around, also help phagocytic cells. And those are dependent on calcium. Okay, so C types. I put it on silent. Okay, but okay. So lectin, right? Lectin type receptors. All right. Calcium-dependent lectins. Okay, those are floating around in the cytoplasm actually, but they will bind to phagocytic cells and trigger phagocytosis. They are like a hook that bind to the pathogen and bring it to the phagocytic cell. And here, the toll-like receptors. So this guy, the toll-like receptors on the surface of the cells. You have toll-like receptors that are going to trigger phagocytosis. And then you have those complement proteins that can also bind to receptors on the surface of a phagocytic cell and trigger phagocytosis. Okay? So here in more details, what are those specifically? Again, lectin receptors, they're calcium dependent, C-type. Important one is that mannose binding receptor. Okay? What are they going to bind? Bacteria, fungi, stuff that is outside of the cell. Your toll-like receptors, TLR2, TLR4, right? And you have a complement proteins, okay? Complement receptors that are going to activate receptors on the surface of your phagocytic cell, okay? The complement floating around PRRs are going to interact with receptors that are called CR1, 3, or 4 on the surface of those pathogenic cells, right? Those are very good at activating phagocytosis. All right, so what is happening here with the phagocytosis? So this was from osmosis, okay? So you have interaction with the PRR, engulfment of your bacteria in this case, okay? And then you are going to bring lysosomes that are here on the surface of on the cytoplasm of this bacteria here. You're going to bring lysosomes that are going to fuse with the phagosome that just ate the thing, and you're going to make this thing that is called a phagolysosome, like right there. Okay, so this thing here is your phagolysosome. So the lysosome that fused with the phagosome, the lysosome brings two types of enzymes that are already present in there because lysosomes are digestive part of the cell, right? So it brings these two types of enzymes. So you have a type of enzyme that is just not blue, okay? So the type of enzymes that are the first ones that come into action, they are ready to work. And they are going to work, they're going to be active at this neutral pH. Okay, so you don't have to do anything to the phagolysosome in order for those enzymes to work. They work at neutral pH. Some of these enzymes are going to start breaking down, trying to kill the bacteria. But, you know, it's not enough. So now you're going to have to activate the enzymes that are azurophilic, they look blue. They are not active at regular pH, but they need an acidic environment for them to work. So this is when your phagolysosome starts to pump hydrogen ions to the inside. This is when you have a hydrogen pump that acidifies the inside. When the phagolysosome is acidic, that is going to activate your azurophilic enzymes, right? So these guys didn't kill the pathogen. Now these guys get activated. So when you put these two type of enzymatic actions together, you're going to kill some pathogens, but not a lot. You're going to kill about 2% of the pathogens that got phagocytosed. That is very inefficient, okay? So that's not enough to kill the pathogens. Only 2% of them get killed like that. So you need a backup system, right? This is when you're going to activate the oxidative burst. Until now, no oxygen was involved. So with those enzymes, both the ones that work at neutral pH and the ones that work at acidic pH, those do not need oxygen, okay? But now you're going to need oxygen. So this is the oxygen-dependent pathway. So this oxidative burst or respiratory burst. So what happens now? You are going to produce nitric oxide, and you're going to produce reactive oxygen species. Okay, you're going to use oxygen, molecular oxygen, to produce reactive oxygen species. Now, what kind? Superoxide. Those are very, very reactive. Okay, they will damage anything they come in contact with. And hydrogen peroxide is also very reactive, but not as bad as superoxide. Okay. Once you produce those reactive oxygen species, now you're really going to burst the microbe, but you're also going to kill the cell, okay? But hey, collateral damage, okay? So going to kill the microbe, but you also kill the cell, so you also have some tissue damage that comes with it. All right, so how is that happening? In order for you to produce those reactive oxygen species, you are going to need an electron carrier. Remember those from biochemistry or cell bio? Yeah. What are the electron carriers? An ADH. Remember? And what else you have there? FADH too. And this guy here, NADPH. This guy here is a very important electron carrier for anabolic processes. It helps you to build up stuff. It's not for catabolic process. Okay, so anabolic stuff uses NADPH. Now, phagocytic cells also use NADPH. So they are going to use NADPH and molecular oxygen. They are going to combine this stuff together and produce nitric oxide. Okay. In order to do that, they need an enzyme. So the enzyme that's going to do that is called inducible nitric oxide synthase. Okay, it's INOS. Okay, so the enzyme is going to make nitric oxide. Okay, so nitric oxide is very, very important to help to destroy pathogens, but is also a vasodilator, right? Nitric oxide is an important vasodilator. Do you want to have vasodilation at the site of inflammation? Yeah. You need to have cells that get there and go through diapedesis, right? You have to have blood vessels that are dilated and also permeable, because the cells have to get there. They have to go to the site of inflammation. Okay, so having nitric oxide there is actually a good idea. Okay, so here in your phagolysosome, you're going to get your NADPH, you're going to get here oxygen, right? You're going to have your reactive oxygen species. So an ADP here because the age was already donated, okay? So you have here your reactive oxygen species, and you know that peroxide is extremely reactive. It's going to kill the stuff, everything very quickly. So you have an enzyme that is going to transform peroxide into hydrogen peroxide. Okay, so reactive oxygen into hydrogen peroxide. The enzyme that does that is called superoxide dismutase. So this thing here is actually superoxide, and you have the enzyme that is going to make hydrogen peroxide out of this. Okay, so this is also toxic, but not as bad as superoxide. Okay, so this is all happening in the phagolysosome. So that takes care of 98% of the stuff that got ingested. Very effective. Okay, and it's also going to kill the cell, but hey, yeah, collateral damage. All right. Now, what is going to happen if you have somebody with a deficiency or with a mutation in the enzyme that makes this nitric oxide, NADPH oxidase? Okay, if you have enzyme deficiency, then you cannot start making your reactive oxygen species, and you cannot go down this pathway of oxidative burst. So what is the percentage of microbes that are going to survive this process if you don't do oxidative burst?
Much higher.
98%. So that's a big problem, okay? Because if you do not have this pathway getting activated because of problems with the enzymes, the pathogen is going to survive. So now your innate cells are going to come up with another type of backup system trying to isolate the pathogen. They're going to start calling on a lot of phagocytic cells to get to the place where the pathogen was not eliminated and start accumulating around that cell. Okay, so here is a cell that phagocytosed, and now you're going to call more phagocytic cells and inflammatory cells to get there and start surrounding your pathogen infected cell in order to isolate it from surrounding tissues. So this is how you end up with a disease that is involving this formation of granulomatomas. So this is chronic granulomatous disease. Okay, so you're going to see this when your phagocytic cells cannot eliminate the pathogen. Chronic granulomatous diseases.
There's no cure for that.
You could use antibiotics. Yeah, because you can also see that in case of tuberculosis, for example. Then if you don't use antibiotics, then you can't get rid of the mycobacteria. Okay. Okay, so here. So that was for your oxidative damage, oxidative pathway, right? And yeah, I only have like 3 minutes for the last end of this lecture. And yeah, I was a bit ambitious putting 60 slides over here. Okay. Now, let's keep in mind here that we're still talking about PRRs, right? And now we are going to talk about this activation of this pathway that is the NF-kappa B. Okay, NF-kappa B activation. What is going to trigger that? Well, extracellular bacteria and viruses and basically all pathogens will trigger this. Okay, so what is going to trigger here? Again, you have toll-like receptors. You have those lectin binding proteins. You have your nod receptors in the cytoplasm, and you have those other receptors that bind to viruses and damage. Okay, so here just a review of how that is binding. Okay, why that is binding. So this is a part that it looks a bit complicated, but is not. Okay, so what is happening over here? You bind the PRR. The PRR is associated to something inside of the cytoplasm. There is this molecule that is called MID88. Okay, this is an association here. binding of the PRR activates response from this guy over here. Then this guy over here is going to send signals to the inside of the cell that are going to activate this protein right here. This protein right here is a kinase. What do kinases do? Phosphorylate something. They put phosphates on stuff. Okay? So this kinase over here is going to do something. First, let's see what's going on inside of the cell before this pathway is activated. You have this guy here, NF-kappa B. NF-kappa B is a transcription factor. The job of NF-kappa B is to go inside of the nucleus, bind to promoters that are going to activate antimicrobial pathways. Okay? So NF-kappa B, the job is inside the nucleus, but it's not in the nucleus. It is in the cytoplasm. Why? Because it's being sequestered in the cytoplasm by an inhibitor. So the inhibitor that is holding on to it, it is called inhibitor of NF-kappa B. That's easy, at least. Okay? The guy is holding NF-kappa B here, is sequestering, is not letting it do its job, is called inhibitor of NF-kappa B or IKB. All right, everything cool. Then you get the signal. Signal transduction. Activation of a kinase. What kind of kinase? Oh, it's inhibitor of kappa B kinase. So this guy is going to go to the inhibitor of kappa B and is going to phosphorylate it. Okay? Here, when you phosphorylate the inhibitor, it is going to dissociate from NF-kappa B. So now this guy is not being held in the cytoplasm anymore. It is going to be free to go in the nucleus and do its job. Okay? So I'm going to stop here. I'm not going to blow up too much of the time. So up to this point, you have PRR activation, signal transduction, activation of the kinase, phosphorylation of inhibitor, freedom of NF-kappa B.